Good evening, everyone. Dr. Meenal Bhujangara Vagmar is here. So, in the series of diseases of field and horticulture crops and their management, today we are going to see the crop that is sunflower. So, our first disease is a Sclerotinia stem rot. Causal organism is Sclerotinia sclerotiorum. Coming to the symptoms, we can find that stem develops the water-soaked spots, which later become converted to the cottony white growth. This kind of some spots will appear first and it will convert it to the whitish cottony growth, and that's why this fungus is appear as a white mold. In this, sudden wilting, that is drying appearance to the flowers and stem and leaf will appear. In this, fruiting structure, that is hard structure, is a sclerotia will produce, which present inside the stem and on the surface. And this sclerotia will survive in a soil or debris for years as a overwinter spore structure. We can see in the disease cycle, there's ascospores are released from the potassium structure, and this lead to the further mycelium growth and infection of the root by this mycelia. And coming that in this typical kind of conidia is not formed. So mainly the cycle will continue by the ascospores and which present inside the apothecium. So our next important disease is Alternaria blight. Causal organism is Alternaria helianthi. In this, dark brown spots with yellow halos and links will form, and later stage this brown spot get necrotic and dry and reduce the photosynthesis rate, and ultimately yield losses will occur. In this, it reduces the size and numbers of seed as well as oil content of the sunflower seed also reduces, and the germination percentage also reduces. Funguses survive inside the infected host tissues. In this disease, basically two important spores are formed, that are chlamydospores. It is a resting spores, and it survives in a soil or debris when host is not available, and when favorable condition with host is available, it will again germinate and continue this disease. Another spores are formed in this disease are typically microconidia, which also helps for the secondary infection spread for this disease. Coming to the next disease, that is rust caused by the Puccinia helianthi, and main symptoms are shown by the rust disease are pustules. Initially, the iridio pustules appear on the younger leaves and later spread over the entire vegetative surface. And this iridia often collapse and covers the large area of the affected plant part. Once iridiospores, the pustules appear on the leaf, that area get dry and necrotic, and photosynthesis rate get reduced and yield losses will occur. The rust cycle is basically led by three spores: pycnospores, ichthyospores, iridiospores, teleospores, and basidiospores. In this, iridiospores is a repeating spores and teleospores is a resting spores. These all spore stages are occurring in the one host, that is itself a single plant, that is it called as an autoecious rust. And these are the 5 stages of spores, and that's why it's called as a macrocyclic rust. Coming to the life cycle, disease cycle, as we can see, pycnospores are here form inside the pycnidia, then lead to the ichthyosporin which present inside the eschia. Then iridiospores are formed, which we are seeing it. We call the iridiospore as a repetitive spores, which can lead a life cycle without depending on any other stages, which further give the teleospores. It is a resting spores, and that lead to the basidiospores. Our another next important disease is the downy mildew, and its causal organism is a Plasmopara held steady. The systemic infection causing the symptoms like stunting and chlorosis along the veins. So, chlorosis will occur inside the leaves, loss of chlorophyll will take place and growth gets stunted due to the reduced rate of photosynthesis. In this, we can see two life cycles are takes place, that is asexual life cycle and sexual life cycle. Asexual life cycle continue by the zoospores and sexual life cycle continue by the oospores, which also act as an resting spores and dormant spore. We can see in the life cycle, antheridium is a male part and a male hyphae, and oogonym is a female gametangia, and antheridium is a male gametangia, lead to the formation of oospores, and that oospores, first before this they form oosphere and then lead to the oospore having thick wall structure, also dormant spore structure. These oospores get germinated and give the sporangium, and the sporangium, after breakdown of sporangium, zoospores are released. These zoospores are having a kidney shell and biflagellated. With the help of this flagella that swims in a water and transfer from one location to another location, and like this it will cause the dissemination. Coming to the management, first cultural practice is a crop rotation. We do the crop rotation to unavailable the host continuous to the pathogen. It will disrupt their disease life cycle. Another cultural practice is the deep plowing. In this, lower surface soil come on the surface. Lower soil come on the surface, and whatever spores or fruiting structure or dormant structure present inside the debris, soil debris, it will kill by the sunlight. Coming to the seed treatment, we provide the seed treatment of thyrum, carboxin and trichoderma. Thyrum, that is 2 gram per kg, and trichoderma 10 gram per kg, and carboxin also 2 to 3 gram per kg we can provide. Coming to the sanitation, we can destroy the infected plant debris to reduce the inoculum. Tillage is nothing but the physical manipulation of the soil which help to speed up the decomposition. We can do this foliar spraying or foliar application of the mancozeb or zineb at the 0.25% of concentration, that is 2.5 gram per liter, and we can provide our first spray when initial symptoms are appear, and second spray after 15 days of first spray or if needed. Like this we have covered the all important diseases of the sunflower, and next we will see the mustard crop. If you have liked this video, please like it, subscribe, and please share it. Thank you.